Alexander Fleming was born on the 6th of August 1881, in the rolling rural countryside of Scotland. He spent his childhood on a farm in Ayrshire, surrounded by wide fields and nature. His family was large and modest, teaching him the value of hard work and curiosity. As a young boy, he spent hours exploring the soil, water, and plants around him. These early explorations quietly planted the seeds for his future fascination with living organisms. Fleming did not immediately pursue a career in the complex world of scientific research. He spent several years working in a standard office job, gaining practical life experience. Eventually, he used a small inheritance to enroll at St. Mary's Hospital in London. It was here that he began his formal training in the field of medicine. He developed a deep, focused interest in the study of bacteria and their behaviors. Fleming became obsessed with understanding exactly how dangerous infections spread through the body. He was deeply frustrated by the limitations of the medical treatments available at that time. Many antiseptics used then were harsh, killing healthy human cells along with the bacteria. Fleming was not your typical rigid scientist. He was known for his unconventional methods. His laboratory was often described as messy, cluttered with various experiments and glass slides. Yet, beneath the clutter, his mind was sharp, observant, and constantly asking difficult questions. He had a unique habit of noticing small details that other researchers frequently ignored. During the chaos of World War I, he worked tirelessly on treating wounded soldiers. He studied how infections took hold in wounds and how the body defended itself. In 1922, he achieved a breakthrough by discovering lysozyme, an enzyme found in human tears. He observed that this natural enzyme could kill certain types of harmful bacteria. This early success fueled his desire to find more natural antibacterial substances in nature. The year 1928 arrived, bringing a moment that would change medical history forever. Fleming returned to his laboratory after a long, refreshing summer vacation away from work. He began sorting through the various petri dishes he had left out on his bench. One dish, in particular, caught his eye as he examined the stack of samples. It had been left exposed to the air and had become contaminated with mold. He leaned in closer to inspect the fuzzy, blue-green growth on the agar surface. To his surprise, he noticed a clear ring around the mold where bacteria died. The bacteria that usually thrived in the dish were completely absent near the mold. Most scientists would have seen this as a ruined experiment and tossed it away. But Fleming was different. He possessed the patience to look closer at the mistake. He paused, holding the dish up to the light, and asked a vital question. "Why are the bacteria dying near this mold?" he wondered aloud in the quiet lab. He carefully identified the mold as a specimen of the Penicillium genus. He realized that the mold was producing a substance that acted as a weapon. This substance was effectively killing the bacteria that were trying to grow nearby. He decided to name this powerful, life-saving substance penicillin. This discovery marked the dawn of a completely new era in modern medicine. The story of this invention is not just about a lucky accident in a lab. It is a story about the power of keen observation and scientific curiosity. The accident only mattered because Fleming chose to investigate it instead of ignoring it. He understood the potential for this mold to change how we treat human illness. However, Fleming faced challenges in developing the substance into a stable, usable drug. He worked as much as he could, but the process required further expert collaboration. Years later, scientists Howard Florey and Ernst Boris Chain joined the effort. Together, they refined the substance, turning it into a reliable, life-saving medicine. Their collaborative work transformed a laboratory curiosity into a global medical miracle. Before this, even the smallest infection could lead to a deadly outcome for patients. Doctors were often helpless, watching as simple wounds turned into life-threatening emergencies. After the arrival of penicillin, surgeries became significantly safer for people everywhere. Infections that were once considered fatal became easily treatable with a simple dose. The world had finally entered the age of the antibiotic, saving millions of lives. In 1945, the scientific community recognized the magnitude of this incredible achievement. Fleming shared the Nobel Prize in Physiology or Medicine with Florey and Chain. His legacy remains a testament to the importance of keeping an open, curious mind. He showed the world that even a messy desk can lead to profound breakthroughs. His life reminds us to look at the world with wonder and persistent inquiry. Sometimes, the greatest discoveries in history are not planned or expected by anyone. They are simply waiting to be noticed by someone brave enough to ask why.